chapter 6. Oh, before we start, everyone here, just an announcement. Next week, unfortunately,
I don't like it. I don't like how we started.
nothing bad happened to me. However, I won't be here in Malaysia. I will be in Brunei next Tuesday. Supposed to give you a class on Tuesday because next week we do not have any practical class. Next week, I'm talking about next week. We do not have practical class, we do not have tutorial, but we only have one lecture. But unfortunately, I won't be here. I will be in Brunei attending a conference. So I'm thinking to give you a live lecture, but I'm afraid there is a risk, right? I do not know my schedule yet. So it's better not to have, and also the Wi-Fi may not work over there. I'm not sure. So if you do not mind, I will make a recording video and then I post it on Thursday, most likely when I'm back. Alright? Okay with you? Okay, so next Tuesday, don't come here. Alright, don't come here. I'm going to post a video recording. It's chapter 7. It's about the virus, about all viruses. And tomorrow we have practical class. Please come to the practical class. If you all have a Wednesday practical class and then Thursday practical class, bring your lab report with you and we're going to mark your lab report on the spot. Okay? Don't tell me. All the time, I don't know my experience last semester, there will be somebody that I forgot to bring my lab report, I forgot my lab report, something like that. But please bring it with you. Okay, let's start. This chapter is about the control of the microbial growth. So, as we know that microbial growth, as long as that is a nutrient, they only need a minimum nutrient to grow. So, but we do not want them to grow, and we call it contamination. So we need to control the contaminations, and this is there for the sake of public health. Therefore, this chapter is talking about how to control the microbial growth. First of all, in the first part, very short, we will talk about the principle of microbial growth. There are some terminology we need to learn. And second part, which is the last chunk about this in this lecture, there will be the method, which is can be categorized into three methods that we use to control. Either we kill the microorganism or we reduce the populations of microorganisms by using these three methods. Alright, the learning outcome. For the first part, you need to actually clearly define what is sterilizations, disinfections, antisepsis, and chemotherapy. And also in the first part, there will be actually when it's putting in the suffix of a word with the cider or static, that is a different meaning. One is killing, the other one is inhibitor. So for the last part, you need to know how to explain how to use the method, what are the methods, and the mechanism. Some of them will have explained to you the mechanism of actions that able to kill or control the populations of microorganisms. Right? So you need to know how to explain that, each of the methods. To start with, there are some terminology you need to learn. Sterilizations, which is considered the most effective way of killing the microorganism, we call sterilization. So how it kills, it actually wipe off everything, all the viable vegetative cells, also including the endospore. Who is the last person to come from this room, this door? So sterilization is the best way to actually remove all the living microorganisms, including endospore. So-called sterilization. Disinfections, I think this is the word that you use most of the time, disinfect or disinfectant that you use. Disinfection is in fact similar like sterilization. It kills all the vegetative cells. The only thing that it cannot do is that it cannot kill the endospore. So it's less, considering it's less effective than the sterilization. Okay, so it go on here. Sanitization. Sanitizations is also, you heard about that, sanitizer that we use during the MCO time. And it is less effective, we consider, and it only able to reduce the populations, but it does not kill the microorganism. Yeah, it can remove most of that to control the growth of the microorganism. Now, antisepsis, I think you also heard about that, but not very often. The first three, right, normally we apply it when we're talking about we are trying to sterilize, disinfect the places, the floor, the object, right? But however, when we use the antisepsis, actually is referring to the living tissue, which is, for example, our skin. So sepsis is microbial contamination. So the entire word is called antimicrobial contaminations. So, and is normally we try to prevent the infection from the microorganism. Therefore, we are using the way of the infections and to apply the chemical, whatever agents, to the animal surface, which is considered as skin, the tissue. Okay. Degerming also similar like antisepsis, but it is less effective than antisepsis. Antisepsis normally seen as disinfection. However, this applied to animate object. This is applying to the living tissue. This degerming is similar like the antisepsis, but is less effective than the antisepsis. Is, for example, the soap that you use to clean your hand, just to physically rub, right? And you also can apply the ethanol on your hand, rubbing it physically, actually removing the microorganism. So it comes with these several terminology, which I think that easily you can distinguish them. And here is the table. I think I taken it from the textbook. So is this a further explanation for each of them? So you can take a time to take a look of it. And this is the graph showing how they actually reduce the populations of microorganisms. So here, y-axis, it could be indicate the number of cells in terms of log, and this is the x-axis is the time. So for example, sterilization is the most effective way of killing the bacteria or microorganism. So in a very short time here, it can reduce the number of microorganisms almost close to zero. As you compare to disinfections, if you want to reach at the same level, right, reductions of the number, it will actually take that much of time. So it is less effective than the sterilizations, and the antisepsis is almost same as the disinfections. It's also taken time to actually 50% of the population wiping off. It take that much of time, but for here it take that much of time. So considering it take longer. However, eventually, if you reach to the same, if you apply it longer, it will the same effectiveness that it takes as the disinfections. Sanitization, I think is between I think it's been around here. It never actually reduce the number of cells down to like 10%. It's always like it reduce the population, but it never go down as good as the disinfection and sanitization. So the number, I think it doesn't give a curve, but I think it's somewhere they retain the populations around here. Not 100%, but drop to could be slightly more than 50%. Okay? chemotherapy, chemo is mean chemical, right? They're using the chemical to treat or kill the microorganism. So it's called chemotherapy. Of course, chemotherapy can use for the cancer patients. Same thing, chemo and then therapy is a treatment. So using the drug to treat the cancer patients. But here is the chemo is chemical to kill the microorganism. And when you actually, the word, sometimes you saw the bacterial site. So actually it's a suffix, we put the site or cider in the adjective. It means it's totally, this is the agent. For example, the agent is bactericide, meaning the agent can kill the bacteria. But if you have something static, or you have example bacteriostat, actually it means that it's the agents can inhibit the growth, but not killing the bacteria. So that is a different meaning when you talk about that bactericide and bacteriostat. Okay, one is killing the bacteria completely, the other one inhibit the growth. Now, we come to the main part, the main content. The method. There are three: mechanical remover, physical control, and chemical control. And we start talking about the physical control. I put it up there because this one is long. So to start, we're talking about physical control method, right? Now, physical control method, they are divided into two parts. One is they're using the heat. Both of them are using the heat, but one is moist heat, the other one is dry heat. And I think you heard about that. For moist heat, pasteurization, definitely you heard about that. And also sterilization. So this is sterilization, as we just learned, is the most effective way of wiping off the microorganism. So it's moist heat belong to that. And for dry heat sterilizations, applying dry heat, and such as putting the microorganism in the oven is considered dry heat. Or UV radiation is also a dry heat. Ionization, radiation, also a dry heat. So we're going to go one by one. So we start with the moist heat. So the moist heat, as what is doing, is definitely using the water. So one of the very standard sterilization using the moist heat is autoclave. Autoclave actually is sometimes you call the machine. The machine need to be used for sterilization. We call it autoclave. But autoclave sometimes it can be used to replace sterilizations. Sometimes it's called we autoclave the plate, meaning that we sterilize the plate. Alright, so but at the same time the machine, the equipment is also not autoclave. Have you seen autoclave before? It's actually everywhere, could be fine. Not in the lab. Normally it can be placed inside the lab, or there is a designated place. I think if you at the level 4, right, there is a lot of practical class, right? A lot of lab. But if all the way you're going to the other side, to the end, there is a storeroom where you can find a lot of freezer. Have you seen that place? Yeah, inside that, behind that you have to open the door, you find a lot of freezer. There is another door, you go in. That is the place that we put the autoclave. So we have a room that we put the autoclave where we normally to sterilize everything that we want to be clean. And if you want to look at it, you can go and take a look how the autoclave look like. Yes, so that is the autoclave. Normally is used in the lab or in the hospital. Because the lab definitely, when working with microorganisms, we need to make sure that the things that we use, at least the equipment and the glassware that has to be autoclave. In hospital, definitely everything has to be clean. So mostly they will actually, even after those contaminated waste, they also sometimes they autoclave it before describe how you actually operate the autoclave and how the step look like. You have 3 steps. So looking at this diagram, first you need to apply steam. It's important. So you have 3 things considered: temperature and the pressure, and also the time here. So when you apply pressure, in high pressure, you actually increase the temperature. So you turn water into steam. So the steam will go here, all around, surrounding it, and you pass through the pressure gauge, which allow you to read what is the pressure. You have to reach certain pressure when you do the autoclave, and then it goes all the way here and enter the chamber. So this is the chamber inside the autoclave. And when you enter it, the first thing you're going to push all the air, the existing air inside the chamber out of it, because this is the cold air, right? So this is not hot. So you have to push it out to here, and then it will go down here. And of course the water will become turned into water and dripping here. There is a bottle to collect the water. And when it's actually pushing, then it will fill up with the very hot steam. So this hot steam can go up to 121 degrees Celsius. And what happens is that you will have the objects. Let's say that you want to actually sterilize the medium. So you have a flask, you have a cap, but normally the cap you loosely, is not tightly capped, but very loosely cap on it, because you cannot just screw it, you cannot use a screw cap. When you use a screw cap, even you use a screw cap, you need to actually turn it loosely. And because you need to allow the steam to go in, because you want to sterilize the medium, so you need to let the steam go in and to get contact with the medium, so that allow it to, the heat reaching the medium and kill whatever the bacterial that in the medium. So you need to have the last loosely cap on the flask. And this is how you do, okay? So it normally take about, the pressure about 15 psi and 121 degrees Celsius for roughly 20 minutes. With these conditions, all the vegetative cells and also the endospore, they all kill. Now, once the sterilization done, 20 minutes gone, then your exhaust phase. Exhaust phase meaning the releasing the pressure. That is automatically this head will be open to release the steam. So when it's released, it starts to cool down. Alright? It starts to cool down, and then once it cool down also take quite some time. It normally take like half an hour. Then you can actually open that and take whatever the things you want to take out of the chamber. However, if you are in a hurry, there is a safety valve which you can manually turn to release more steam out of here. So that is the way. Normally we cannot wait, and we just open that. And of course, in the emergency, you can also open this to release the steam out of here to take the things out. Alright, so this is the autoclave. So this is what I mentioned. By standard, normally in the lab, in future, I don't think you have the chance to actually do the autoclave, because normally it's done by the lab technician, and but you know where you put the stuff and allow the technician to do the autoclave for you. So this is a condition normally it's been set, 121 degrees Celsius. Normally I take 20 minutes instead of 15 minutes, and the pressure is 15 pounds per square inches. So when you have the volume of steam keep constant, if of course the machine inside the chamber, the volume of steam is constant, then the temperature will be proportional to the pressure. So that is a table indicate when you're increasing the pressure, you actually increase the temperature in proportion. Alright? Yeah. But the next slide will show you different microorganisms. How do they, like for example here, this table showing you different microorganisms, the requirements slightly different. For bacteria, if this is a vegetative cells, talking about the cells, not endospore, 10 minutes, 60 to 70 degrees Celsius will be sufficient to kill everything, the vegetative cells. However, when we consider the bacteria may also in the form of endospore, therefore, to kill the endospore, we need to reach 121 degrees Celsius for at least 12 minutes. Right? It depends on the type of endospore. So it's between 0.5 to 12 minutes. And for yeast, it's easier. Is like because yeast has cell wall, no cell wall, right? So yeast taking is just 5 minutes, 50 to 60 degrees Celsius. And for the spore, it's 5 minutes, 70 to 80 degrees Celsius. Easier, lower, the temperature is lower, doesn't reach 100 degrees Celsius. For the modes, it's also similarly like this, but the time take longer. Virus, of course, virus basically doesn't have a cell wall. It's only make the envelope is all the protein. So all protein easily denature. So it's 30 minutes, 60 degrees Celsius. That's sufficient to kill all the viruses. Now, this is how the heat apply and actually mainly denature the protein. So protein has its structure. Protein has its structure, and the structure has to maintain as its proper 3D structure in order to function. So when you apply heat, basically in 30, in 60 degree, is already denature the protein like this. So they become linearized. And if you further apply heat, then they will start to coagulate with each other. Once they coagulate with each other, there is no ways back. So it totally destroyed all the protein in the cells, and you kill the microorganism. So the heat and actually denature and then coagulate the protein. Alright. Now, how do we know that whatever you put the glass or whatever the bottle you put inside the autoclave, this is the autoclave, a very small autoclave. You put inside is like has been sterilized. Can you make sure? We are not too sure, right? We just know that we operate it, but whether that is the temperature reached as we expected or not, we do not know. So we need some indicator to indicate that the sterilizations work. So this is a very common because it's just a tape and it's not expensive. So it's called, we call normally autoclave tape. It has a stripe. So the stripe in white color, right? And the stripe is actually heat sensitive. So once we tape it on the glasses everywhere when we want to indicate, alright? So each of them we have the tape. So once it has been autoclaved, then we can see the stripe become black. So that's indicate it worked. However, this tape is actually outside the bottle. How we sure that inside the bottle is actually effectively also reaching the temperature and also killing the bacteria inside the bottle. But normally we believe that whenever we tape on it, it works. In the chamber, it should able to remove everything from the bottle, unless you actually screw it very tightly. The steam cannot go in. Therefore, you cannot actually sterilize whatever inside. So make sure that in future, whenever you try to sterilize your bottle, your screw have to release the cap slightly. Okay. So to ensure, there's another way, but it's slightly more expensive, that you can ensure that everything you put inside, including the endospore, will be killed. So it's called the biological autoclave indicator. They are selling the product in this way. Let's take an example of this kind of the tube. So inside the tube, they actually put a biological indicator, meaning they're putting this bacteria, the spore. They're putting the spore inside the tube. So this spore come from dual bacillus sterile thermophilus. So that is a microorganism, gram-positive bacterial, rock shape. They are thermophile, meaning that they can endure high heat and then heat resistant endosperm. So normal, below 100 degrees Celsius, you may not be able to remove the endosperm. Now, they're putting the spore inside, and they also have the indicator, meaning the pH indicator, so it's purple color. Then they put this tube together with whatever you want to autoclave into the autoclave, the machine. When it is done, you take the tube out. It's not finished yet. You take the tube out. Suppose that you put this tube containing the spore into the autoclave. The spore will be killed, right? So when you take it out, you put it at 37 degrees Celsius, maybe an incubator, maybe the heat block with 37 degrees Celsius, to grow them 24 hours. The next day, there's nothing happened because the spore has been killed. However, if you have the color, yellow color, turning like that, indicate there are something wrong. So you may see the color change from purple to yellow. The yellow is also a pH indicator. When it is acidic, it's yellow. And acidic is because the bacteria produce acidic product. And also you see the turbidity. There are two things you will see from tube: color change and also the turbidity. There's something wrong. So you fail, indicate you fail to kill all the spore inside the autoclave. However, this way more expensive. Buying this, and I'd rather use the tape. Now, we come to the pasteurization. Pasteurizations is also a moist heat sterilizations. So is pasteurization come from the person called Louis Pasteur, right? You know him, everyone know him. And he actually created, we're still using the concept of pasteurizations in the daily industry. So but it's different from sterilization because the origin principle of pasteurization is to actually heat, right? Heat, for example, the milk at below 100 degrees Celsius, but for a longer time. So is it 63 degrees Celsius for 30 minutes, and then immediately you cool down it to 4 degrees Celsius. This is pasteurizations. Why they are not heating that into 100 degrees Celsius? Because for food, we need to actually keep the texture of food, the taste of food, we keep it as original as possible. So we don't want to change the taste of food. Therefore, they cannot actually heat the food up to 100 degrees Celsius. Don't worry. So this is the pasteurization. Now, we have a new way to do the pasteurizations, which is called ultra-high temperature pasteurizations. Have you seen this ultra-high, right? You found it. Now the milk, you don't need to put with the cold temperature. You can put on the shelf, right? They can even, but once you open it, you need to keep in the fridge. But if you not open it, you can keep on the shelf at room temperature for 3 to 4 months, no problems. And because those milk has using this way for sterilizations, and this is a way you different. They may slightly different, but in general, they were heating the milk to 135 or to 150 degrees Celsius for a very short time. Okay, for 1 or 2 seconds, and immediately they cool it down to room temperature and further to 4 degrees. And on top of that, they also do this one UHT in a closed system, meaning that they heat up to sterilize the milk. Same time, before they moving that out, in the closed system, everything closed. So inside this closed system, everything is sterilized. They do the packaging, they seal the package. So nothing, so it's just to make sure that the quality of the milk. So no contamination. And so that's why it's now getting popular. So you can see pasteurization, the method, they will call it pasteurized recombined milk. And the package for the UHT is slightly different. They will be like this, and they will indicate UHT milk. So always remember, pasteurizations as below 100 degrees Celsius for a long time, at least 30 minutes. But for the UHT, you apply with a very high heat over 130 degrees Celsius for a very short time, and then immediately cool it down to 4 degrees Celsius. Now, dry heat. What time is it? I saw it. Just to wake you up. Now I just see the time. When should I have a break? Not yet, right? Not yet. Let me finish. Dry heat. Using the heat, but this is a dry heat, no water involved. So dry heat is less effective. Imagine that dry heat is like an oven. For example, oven also provide hot air. You can increase the heat inside the oven 170 degrees Celsius, but it would require 2 hours. So it's compared to autoclave, same effectiveness can be achieved if you only apply 120 degrees Celsius with 20 minutes. So that's the difference. You take longer time, higher temperature, using the dry heat to kill the microorganism. But dry heat can also ensure that can completely wipe off the microorganism, such as incinerations. So incinerations, I think each large scale, normally incineration is required when we try to actually for the medical waste, because we make sure that, for example, coronavirus, right, in the Wonder Pack, all the coronavirus will be contaminated. You want to make sure you totally kill the virus. You actually go put them for the specialists will come to collect it, and for the incinerations. So incinerations is something look like this. You're going to truck, you bring all the waste coming here, then they will go through to break it down into small particle, and then come this combustion chamber. They apply the temperature very high. I think most of the time is over 1000 degrees Celsius. So it totally can actually, even the metal can be melted at this temperature. So at the end, it will actually generate ashes. So the ashes can be recycled to use for different purposes. For example, to use to make the brick, or to actually fill the land. But incineration has one issue. Is normally with this combustion, they generate a lot of toxic gas. So the toxic gas, for example, dioxin is one of the toxic gas that actually they have proven that is carcinogenic. So anything gases coming from this combustion chamber, they will have to go for treatment to remove all the toxic gas. So whatever come out from the chimney must be non-toxic. All right. So very expensive, but this way incineration ensure everything killed. And here, flaming the loop, you done this, is also considered dry heat. Alright, oven is considered dry heat. Alright, next one, UV radiations. Everyone familiar UV radiation? You might have one at home, right? No? You could have one at home. It's not something that expensive to be like. I learned from YouTube they can use the smartphone, the handphone to make the UV light. But I do not know how to do. But if you want to try, you can use your smartphone how to make the UV light. Now, UV light actually, if you put 260 nanometer, these are the wavelength. All right, by this wavelength, the absorbance is the best is for the DNA. So DNA absorbs the UV light at 260 nanometer. So if you you want to kill the bacteria and you want to destroy the DNA, that's the best way to destroy the bacteria, to destroy DNA. You put it at 260 nanometer. So with that, the UV light, how it killed the bacteria by actually dimerize the diamine. So the diamine, if you have two diamine T and T sitting next to each other, then you will have forming a dimer under the UV light. So it basically, it doesn't cause any trouble, but it could cause trouble in transcriptions, but the most trouble it causes is during the next replication, the next cell division. You need to replicate the DNA. So if you have a forming a dimer, sometimes you consider, the DNA polymerase consider is a single nucleotide. So when it's replicate, you consider one nucleotide is instead of AA, you just put a single A. At the end, you will have a deletion in your DNA and mutation. The mutation will make a lot of mutation in different genes that make the bacteria die. Okay? So this is UV light, how it kills. So UV light normally apply on this. I'm sure you have seen this, the biosafety cabinet is in the lab. Every single lab we have biosafety cabinet. So before you use the biosafety cabinet, normally they have procedure. Because you want it clean when you use it, you turn on the UV light for 15 minutes, 10 to 15 minutes, just to make sure that everything inside is clean, is killed, because it can kill effectively. And CO2 incubator normally we keep to culture the human cell line. So it doesn't turn on all the time, because incubator always we use it, but we need to clean the CO2 incubator from time to time. When we clean it routinely, we turn on the UV light. Okay? Oh, have you seen this? Oh? For the baby, you have baby at home. So you know this is for the baby, right? You've seen it before. Yeah, it is for the baby, that's right. This normally, that's why I say you can find this, something like this at a household item. And so this is for normally, because we need to keep, baby is very fragile, we need to keep everything clean. So you put everything for the baby, whatever they use, not only the bottle, but also everything that the toy, you can put it to sterilize. So very convenient, and it's not expensive, I think. Second one. What is this? It's likely to be used in the industry. I also have never seen that.
Water disinfection.
Water disinfections. You see what you say? Yes, that's correct. This is for the water. The water, if you want to have a clean water, really, really clean, you definitely have a lot of filter, right? Before that. But finally, when you come to here, after you filter, but you want to make sure that 100% remove the microorganism, you will passing through this tube. Actually, it can be very large. It depends on the scale of the water system. So this is the tube, will emit the UV light, and the water will run through here. So when the water running through, actually exposed to UV light to make the water clean. Okay, this is how is the water disinfectants. And the last one, I think I've seen that before. This one you can buy one to put it in your house. You make sure everything clean. So this is something portable. You can move them around. So this is a UV light. So it's to make sure that you put it in the room and expose it in UV light and make the room clean. It is normally used in the hospital. I have seen that in hospital, especially in the surgical room. They have this all the time. So even that they put in the toilet, you see, and also in all those hospital. All right. dry heat. The last one is the ionizing radiation. Different from UV radiation, ionizing radiation is more powerful. How powerful is it? They have a few of that, and normally the one that we use to actually for sterilize is the gamma. So except alpha, or the UV light, UV light cannot pass through the paper. So if you have the paper, normally when you actually to clean your equipment, right, the surgical tool, you actually expose directly under the UV light without anything, not even putting something, a bag, because even the paper can block the UV light. UV light cannot penetrate a paper. However, gamma radiation, X-ray, all this can pass through the paper. And if you have a thin wood or thin aluminum, it still can pass through. But the only thing that it cannot penetrate is the very thick metal plate. Alright, it cannot pass. So normally we use gamma radiation for the sterilization. It usually used for the food industry. So this gamma radiation is just like the radiations that even human, we cannot expose to that. If we expose to that, probably we will have causing us to have cancer, because the gamma, any kind of radiation, is causing the break of the DNA backbone. DNA backbone contains sugar phosphide backbone, normally we call that. If UV radiation, you only destroy the bases, the diamine, so it can be repaired. But if you actually break the backbone of DNA, it cannot be repaired. So the damage is immediate and very effective. So even we ourselves cannot expose to gamma radiations. Alright, and how it also it generates a lot of free radical. You know that free radical is very reactive. You will actually steal the electron from anything that they in contact. So tissue can be damaged by the free radicals. And this is how they use to sterilize the food product. So this is the way. And a lot of food right now we using this gamma radiation because it doesn't require easy. And you can actually put, for example, we have putting them in the conveyor belt, right? Letting it to go to the gamma radiation, so they will expose it on the conveyor belt, and you do not need to actually unpack. You can put the fruit in the box and passing through the belt, no problems, because the gamma radiation can pass through, can penetrate the cardboard. And what else? So those are the normal food that they use for gamma radiations for sterilization. Fruit, vegetable, chicken, turkey, whatever. And shellfish, spice and seasoning, shell egg. And this is a logo that is an international logo, indicate the fruit has been sterilized by gamma radiation. Other than food, what else? All this, such as those medical material, and for example the medium here we cannot put into the autoclave because the medium contain, for example, the growth factor. The growth factor, if you destroy by autoclave, no longer the growth factor can be used to support the growth, right? So they cannot go through sterilization, so they can only use the gamma radiation. And this one is the filter. The filter normally see in later we will see this filter. The filter you also can use for exposed to gamma radiation, because the filter is a filter paper inside. You cannot put it into moist heat because it will be wetting the filter paper, which is not ideally is not good. Do you want to have a break? Yeah, I think actually more than 40% in this room actually fell asleep. Nancy is still sleeping. All right, you can have. I give you 10-minute break. Yeah, you can do whatever. Buy some coffee and drink.
Another ones. I saw your name. Come on. Luis, 方圆，你还没有 check in 啊？
check 了吗？
还没有，我刚说你。 懂吗？你们吃啥啊？啊。还有一个什么？这里一个。都比较屌。
有选夹吗？是哦。我给你sample 啊。
我那个。
我几个？那你过海关啊，过刚上飞就我了，是吗？
你知道吗？我的脸真够了，好吧，随便。滚，我被人。
See your classroom. Alright, everyone's back. Okay, let's start. The next one is the mechanical remover method. So it's mechanically removing the method. What is the best is the one that we have here, the filtrations, right? So normally a lot of water purifications can use the filter to remove the contaminant. So this is one way, the filter. And we use very often in the lab as well, and also to purify water. And how does it work? Basically, the filter has a very, very small hole. So it's having a very small hole, allow the filtrate or the water to pass through, but it will trap all the microorganisms right on the top of it. With that, the filtrate will be a clean fluid or the water. So there are many different types of the filter. They can be in different size, and also it's different purposes. This is for air or gas, and this is for solvent, for the aqueous solution. So there are two types at least. Also, they have, even for solvent and aqueous, sometimes they have their specific feature. For example, they have some low protein binding, so that it doesn't trap the protein, and also is used for the micro and for different purposes. All right. And this is how they use for the aqueous filtrations. So normally we have the filter, which is here. So if you want to sterilize the medium, this is a medium red color. Normally this is a medium for the human cell line. So we normally not to autoclave because it's contain a lot of like growth factor, protein that essential for the growth. So we cannot sterilize it with the autoclave. We use the filtration. So the filter here, we're just using the water to suck up the filter. So this is a filtrate which is sterilized, and we can use to culture the human cells. And for easy one, this is a small one. Normally it's attached to a syringe. So then we pass through whatever the things, the solution we want to sterilize. We pass it through it, so it comes into here to collect those who are no contaminations. So this is contact to the syringe. And if you can see, there are two types. One even the pore site. Normally when you purchase the filter, you look at the pore site. This is 0.22 micrometer. So the pore site, the diameter is 0.22. And normally this one is good to filter, to filter the microorganism, especially the bacterial. So bacterial is bigger than this size. So we never use to filter the bacteria with 0.45 because I think the bacteria is around 0.4 to 0.3 micrometer in diameter, most of them. So we need to use the right filter to filter whatever the microorganism. Here, this is the called mini-Q water, which actually all lab have this mini-Q water. Have you seen that? The water coming out, they have two tanks. This is one tank which already gone through one filter. The filter is like a column. So they pass through the column, actually they filter. Inside there's another filter to filter it. But this one is just the first stage. Then you go into here. So you actually go into a several filter here. Eventually it come into here. And there is another filter here. So then they have a different, the hole of the filter, different pore size, actually can filter out different microorganism or even particle. So as small as this, they can actually filter the metal, iron, potassium, sodium, magnesium, they can filter that out. And you get out of here, what happened is that you got 100% H2O. That's it. Very pure water, just having an H2O without any mineral. And please do not drink this water because it's not healthy without any mineral. You got only the water, that's it. Alright? But normally we use it to prepare the solutions. And sometimes even without autoclave it, we can directly use it in culturing. So this is a very good system, but very expensive. And normally we just simply call it the sterile water coming out from here. We call it mini-Q water because so famous, the company selling this mini-Q water. Alright, now this is another type of filter, but it's filtering the air or the gas. Example is surgical mask. The one that you're wearing is an air filtration. And for example, there is a classic way when you grow the culture, you put a cotton, right, on here, and then it's actually the cotton can filter like the big size of the microbe. So it is another kind of very simple filtration. However, very advanced, we use this kind of laminar flow hood or so-called, forget about class 2, normally they are class 2 biosafety cabinet. So you see a lot, I think you can see the laminar flow hood. And as well as we have many biosafety cabinet in all lab in Sunway. And they are not cheap, they're expensive. But they have a different name. They look similar, but different. This of course is more expensive. And this is having a simple function. Let us see, they serve for different purposes. So that laminar flow hood is used to prepare reagent, medium that you need to culture, just for preparation of medium. You do not bring your microorganism and work with the laminar flow hood. Ideally, no. If you have microorganism you want to work with, you want to keep it clean, not be contaminated, you work with the biosafety cabinet. So what happened to laminar flow hood? Why ideally is not used for microorganism? Is you look at that, there is a filter called HEPA filter, which is placing here on top of the chamber. So what happens is that all the air coming in will go through this. Actually, this one is stirring, and then you actually go through this HEPA filter and come into here, inside. Now, they have something to blow the air through this direction. So the air is blowing out of the hood. So if you are sitting here, So actually the air is directly hit to your face. So you cannot handle any micron while using the laminar flow hood because the air is rushing out. So this one is only used for preparations of the medium. Alright? However, the biosafety cabinet you can use to working with sunlight, organism, even sometimes tissue animals, small animals. What happens is that how is the biosafety cabinet work? So there are two HEPA filter. One is placing here, similar like this. So you have the air, the dirty air come in. So it will go through the filter. So the filter come down here, actually is clean. However, the filter will not actually coming out through this because you are sitting in front of it. So it wouldn't come out. It will actually recycle and it suck it up at the back. So by sucking up, it's a purple arrow. This purple arrow actually sucking up the air. So the air will go to here. There is another HEPA filter. So that it will trap anything, the microorganism will be trapped with the HEPA filter. And then whatever it coming out from the cabinet, it will be a clean air. So biosafety cabinet ensure go in, the air is clean. Whatever coming out, the air is clean. There is no contaminations to the environment. So ideally, this is something that you can work with the living tissue or living organism. Okay, clear? Now, we come to another different. This is using the chemical. Maybe it's a chemotherapy. It's a chemical agents to control the growth. And we see it all the time because, like for example, this is the first one. The chemical is phenolic. Phenolic is also can call it phenol. Normally it's phenol, and also can be called carbolic acid. And has been used by a British surgeon first, Joseph Lister, and I think he's a surgeon for the treat the surgical wound. All right. Of course, now we're no longer using this phenol to treat the wound. And however, since the discovery of this phenolic compound, there are a few of this. They has been using in the mouthwash. Listerine is named after Joseph Lister. You see that? And you will look at this in the ingredient. They contain dimer. Dimer is a phenol. So it's a phenolic compound. And also strepsil that we have all the time. We also can find one of this phenolic compound. What is called amimethylcrystal. Okay, it's present in the strepsil. So this allows them to actually disinfect whatever in our mouth. But for sure that it is not very effectively killing all the bacteria in your mouth, but it at least can reduce the growth, inhibit the growth, and reduce the number of the bacteria in the mouth. And how it actually work? It can against both gram-positive and gram-negative bacteria, but based on this paper, I think this diagram and how it works, the phenolic compound have one aromatic ring and also have hydroxyl group. The most simple, the phenol, have these two. And basically, they can interrupting the enzymatic activity by hydroxyl group can make hydrogen bond with protein, so that is interfering the enzymatic activity. Also, because this aromatic ring is a very flat plate, so it can intercalate into the DNA and disrupting, changing the DNA structure. Because of the plate, it's very thin and very flat. And again, this aromatic ring is also altogether is also non-hydrophilic. It's non-hydrophilic. It's hydrophobic. So it can go into the membrane and disrupt the membrane. All right. And finally, it can also the hydroxyl group can also disrupt the ribosomal translations with interacting with the one of the I think it means the RNA of the ribosome. So it has several ways they can actually interrupting their bacterial activity. Now the second chemical here is a heavy metal, but we are not using a lot of this heavy metal because heavy metal we also not but also very toxic to human too. But I'm trying to find any whatever the current things, equipment, whatever we use heavy metal. One that I can find is the CO2 incubator. Not all the company are selling this kind of incubator. And what is special for this incubator is that they have, you know, the inner wall, the inside, that is the wall actually is made with the heavy metal, copper. Is it copper? Yeah, 100% pure copper, antimicrobial interior. So actually it is fun. So therefore, this incubator that the bacteria is very difficult to attach on the surface inside the chamber and is inhibit any growth of the bacteria by using the copper, right? But it's also it's not ideally not many of the companies selling this type of incubator because the heavy metal is also not very good for human as well. This is a question asking which is more resistant to disinfectant, gram positive or gram negative. You know that gram positive like this, the cell wall and membrane. Gram negative have outer membrane, cell wall, inner membrane. Which one? Gram positive, more resistant. Why? But in actual fact, they have done it, and the actual fact, they are more resistant to the disinfectant is the gram negative because they have this lipopolysaccharide, right? And also some lipopolysaccharide and some protein actually in the outer membrane. So they can actually block any antimicrobial agents to come into the cells. Especially we're using a lot of antimicrobial agents that purposely targeting on the cell wall because humans have no cell wall. So we use something that the agent can target on the cell wall, very specifically kill the bacteria. And because of the membrane, so they are not easily accessed to the cell wall, unlike the gram positive. Alright, it's just like antibiotic. Yeah? Now, we talk about halogen. Halogens in you found halogen in your what table? Periodic table. Which group? How do you know? Did I put the 17? You know. Okay, good. You just know. Everyone know. Sorry, I need to check before I come here so that I know it's group 17. Halogens, so there is a group 17 is all halogens. Then I asked this group 17 had contained iodine, chlorine. I think it's come with the fluorine, chlorine, bromine, iodine, and then I read it but I forgot what is the elements called. There are another two, alright, which we don't see that very often. So this is halogen. Why the halogen is a very effective antimicrobial agent? Because halogens normally appear as a form of gas. It's very active. it's very reactive. It can reactive that interacting with a lot of like organic materials. For example, we have been using iodine. You definitely hear about, you've seen this one, right? We're selling in the chemist. So you have the betadine. Betadine is for the mouthwash. So this mouthwash is contained, if you look at the povidone iodine. So it contains the iodine in the mouthwash. And basically, if you look at iodine, it reacts to everything, DNA, fatty acid, or even anything that interacting with them. And of course, betadine, using selling this one is very popular. Povidone iodine, which is something we put on our wound. If you have a cut, something, a wound, that normally we apply that. Anyone using this? I have asked this question. Malaysian people definitely know what is that. What about other country? To anyone that doesn't know about this one, we use it when we were small, a kid, when we actually has injuries. We are still using it, medical doctor is still using it to actually apply it on the wound. Alright? So, chlorine. Chlorine is also very reactive, and it is a gas can mix with the water. When it's mixed with the water, and it becomes an oxidant called hypochlorous acid. So this hypochlorous acid, what happens is that when it turns to hypochlorous acid, it can damage the membrane. It can actually interfering with the protein folding. So the protein become not folding properly, and you will aggregate, all right, during the protein synthesis. So, it actually damaging DNA as well as the protein. And with that, so therefore, it actually can be used as one of the antimicrobial agents. So where did you find this chlorine? The chlorine as a form as a salt, because it can react with salt. It's a salt lover, so they can make it into hypochlorite salt, which is normally present in one of the Clorox as well. But also one of those are used for clean the swimming pool. But however, it has something not good, because we normally like Clorox, right? You never drink Clorox, right? You don't even actually use it for shower, right? So it's very irritating to the skin, nose, and eye. Alright? So that's not something not good. But it is very effective to kill the bacterial cells, vegetative cells, and fungus, but it's less effective to some of those like Cryptosporidium. Cryptosporidium is somehow like the Plasmodium. Same category as the Plasmodium, the one that causes the malaria. So this Cryptosporidium had a very hard shell, which not allowed this hypochlorous acid to penetrate. So if you have this hard shell, it can protect. So it's not 100% very effective. And I'm going back. Fluoride. Fluoride. We have the toothpaste. Did you actually look at the toothpaste that is contained fluoride? If you look at this one, you see sodium fluoride, 0.32%. But nowadays, I try to check, my toothpaste don't have the fluoride. They don't use it anymore. Because that is the research indicate that the fluoride also not good for human. When you take it constantly, it's a long-term effect. Yeah, it will be harmful to human as well. So if you look at the toothpaste nowadays, not many toothpaste, not many brands are still using the fluoride, contain the fluoride inside the toothpaste. But what is good that they are used to be very, very popular to be incorporated in the toothpaste, because it actually can remineralize we have the coating on our teeth, right? It's called the enamel, right? Enamel is something coating on the tea. And inside the enamel, it contains so-called, it's a crystal, sort of like a crystal particle called hydroxy apatite. So when this hydroxy apatite is exposed to acid, actually it will be like melted. So if you have plaque, the biofilm grow on the tea, so the biofilm produce a lot of acid, so it can actually make the tea decay. Now, if you apply the fluoride constantly, like in the toothpaste, actually the fluoride help to fill up the hole or the gap, whatever, to remineralize the enamel. So this is something good for the from the chloride, which will make the tea stronger and protect from further decay. But you check your toothpaste. Do they still have fluoride in the ingredient? I don't think it have anymore. Not many have it. Now, be closer to the end. Yes. This is the alcohol. Everyone familiar? No need to explain. Alright? You use alcohol 75% or 70% or 80%, which is fine. So alcohol can effectively kill the bacteria and even the fungus. What else should I explain? Since you're all familiar. But remember, we use alcohol with 70%. Why not 100%? This is one thing. But with water, it kills better. If you can do a test, you just put, you not consider evaporation, you actually not keep it evaporate. But with 70% with the water, 30% water, comparing the 0% water, this 70% ethanol kill better. Why? I think I will keep it for your tutorial, alright? Alkylating agents. Alkylating agents, normally you will not have it because it is a control chemical. You will see only that in the lab. Formalin. Do you use formalin? The formalin normally is used for the, what is that up? To put the, for fixation. For example, you have the specimen, you want to keep it like a snake, so you put into the bottle, you put it there, you can keep it for a long time. Alright? For example, here, normally we use it because it's very effective. It also can kill a lot of things. Any organic material, including any kind of microorganism, even human, can be fixed or killed by the alkylating agent. It just depends on how much the concentration we apply. Of course, if we say, for example, you can normally in a concentration 37%, they are selling with the bottle, it's 37% on, called it formalin, right? You can actually wipe on the bench, on the floor, and especially they use this one, the butyraldehyde is a 2% in this particular bottle, normally used in the hospital to clean the surgical and medical equipment. And this one, they actually to decontaminate the biosafety cabinet. Of course, normally we keep the biosafety cabinet clean, we apply the UV light, we wipe with the ethanol, and normally they not recommend to wipe with ethanol. So we clean it constantly, but once a year or twice a year, you will ask somebody to come over to use the gas. I think they used to get what kind of gas? Formalin gas. How do they wrap the whole cabinet and then they insert to actually apply the gas inside this, and then the gas will go everywhere because the gas can go everywhere inside the cabinet. So they buy that. I think they leave it for half a day. After that, then they all very, very clean. So you need to ask the company people to come here to do that. And if you have seen that, I think I've seen once. actually is very powerful agents. Alright, this is the last slide. There are some factors. It doesn't mean that using different chemical agents and this chemical agent is better than the other. Even you use the same chemical agents but you apply in different microorganisms, the effect will be different because it's all depend on this factor. For example, the population size. Of course, if you have a large area that you want to actually disinfect, you need to have a longer time because it will be cover more of the microorganism in the larger area, right? And the population compositions, for example, what kind of composition? If the biofilm contain one type of bacteria easy to kill, or it contain heterogeneous different type of bacteria and also contain endospore, that is totally different story. If it contain endospore, you to apply more harsh agents to kill the endospore. So in populations of the biofilm is depends on what is inside. Concentrations, of course, higher concentration can kill the microorganism better. However, concentration we need to control to a standard, public health standard, so that it's not being harmful to human. Duration of exposure, definitely longer exposure kill more microorganism. So you make sure that when you actually clean your house using the bleach, like Clorox, normally you can clean the floor, make sure that after you wipe, you leave it for at least 5-10 minutes. Right? Then you actually clean with the water. So longer exposure time help you to kill better. Temperature, higher temperature increase killing. A lot of chemical agents, when it is slightly in higher temperature, let's say over 30 degrees, you apply the chemical agent, it works better. If you actually reduce the temperature, like 10 degrees Celsius, it works less effective. So higher temperature actually can help the chemical agent to work better, normally in this way. Also consider the local environment, whatever the pH of the biofilm, and also the complexity of the biofilm, right? And also take into considerations how you're going to apply the chemical agents. So these are the factors you need to consider when you apply chemical agents. It's not saying that this chemical agent is very effective. Even this alkylating agent, you kill 100% of all the microorganisms, but if you do not do it properly, you do not control the exposure time, do not control the concentration, it may not work. Okay? All of this. All right then, that's all.